Welcome to session 2 of Bioinformatics. I am Dr. Y.V. Rajesh, Assistant Professor, Department of Biotechnology, Koneru Lakshmiyya Education Foundation. In this session 2, we are going to learn about applications of bioinformatics. So in session 1, we defined bioinformatics as a multidisciplinary field which conceptualizes biology in terms of molecules and applying information techniques to understand and organize huge amount of data here. So we continue that session. So the aim of this session is to explore the diverse applications of bioinformatics in scientific research and industry and instructional objectives here. So here we try to identify the key applications of bioinformatics and how bioinformatic tools are applied in different areas of life science, and what is the impact of bioinformatics on the advancements of biological sciences. So by the end of the sessions, students able to recognize the role of bioinformatics in genomics, proteomics, medicine, agricultural sciences, microbial taxonomy here. So here also students able to illustrate how bioinformatics will contribute to personalized medicine and drug discovery here. So we focus on its applications in microbial genomics, medicine, agriculture, animal husbandry, followed by other fields here. So the applications of bioinformatics can be attributed to what is done through bioinformatic algorithms here. So basically three kinds of analysis is done in bioinformatics: structure analysis, and second analysis we do is sequence analysis, and third analysis done in functional analysis. So structure analysis involves understanding the structure of nucleic acids and develop algorithms to predict its structure from the sequence. And protein structure prediction, it involves design algorithms based upon experimental data and use them to predict secondary and tertiary structures of the proteins. So based on the structures, you try to classify them into various families or structural classifications based upon various classes, architectures or topologies here, and then compare how different structures are linked to each other. And genome sequence analysis, or simply sequence analysis, it involves genome comparison, phylogenetic tree constructions, predictions of functional regions such as genes and promoters, and develop algorithms for the motifs and design novel algorithms for database searching and perform sequence alignment. And function analysis, third kind of analysis, it involves metabolic pathway modeling. So if you take a simplest organism such as E. coli, you have 200 metabolic pathways are there, and complex organisms such as human being, you have thousands of metabolic pathways are there. So we can do metabolic pathway modeling and understand gene expression profiling, protein interaction predictions, protein subcellular localization predictions here. So various kinds of analysis can be done in bioinformatics in different fields. So based upon the analysis, the outcome will be different for each type of analysis here. So in structure analysis, basically a primary sequence of protein, linear arrangement of amino acids linked through peptide bond can be used to predict three-dimensional structure where alpha helices, beta sheets are connected to hydrogen bonds or van der Waals interactions to form functional proteins here. And using this functional protein, if it is a pathogenic protein or involved in a disease, so we can find out a drug or a ligand molecule which will go and bind to the protein to modify its activity so that pathogenicity is reduced here. In function analysis, so in sequence analysis, As I told you, compare sequences. It can be a gene sequence or protein sequences from different organisms using sequence alignment algorithms, multiple sequence alignment algorithms, to derive or to find homology among the sequences, determine origins of the sequences, find new specific sequences, construct evolutionary trees here. So in this example, 5 protein sequences which correspond to human hemoglobin, mouse hemoglobin, chimpanzee hemoglobin, rat hemoglobin and cow hemoglobin are taken, and they are analyzed through multiple sequence alignment algorithms to understand how these organisms and how these proteins are linked together. So in this sequence, if you try to understand, here you can define the positions of the aligned sequences into conservative sequences and non-conservative sequences. So conservative sequences, they doesn't undergo much change, whereas non-conservative, they undergo change here. So here K, which indicates lysine, alanine, proline, the position 1, 2, 3, 5, 6, 7, 8, these positions are same, or the amino acids present in these positions of the protein sequence in different organisms is same, and those are called as conservative sequences. Whereas if you take this position, the position which is not scaled by gray background, here you have 2 serines and 3 alanines, and in this case you have 2 threonines and 3 serines, 2 leucines and 3 prolines. So these are non-conservative sequences. So you can use the information of non-conservative sequences and align the organisms. So you can see human and chimpanzee are more closely linked together, mouse and rat, they are more linked together, and compared to all these species, cow hemoglobin sequence is an outlier, slightly outlier compared to all these protein sequences. With simple sequence alignments, you can derive insights such as tree constructions, specific sequences, conservative positions, and finding homology. And coming to the function analysis, so you can compare gene expression among different cells here. So this diagram is actually a simple microarray experiment. Microarray experiment, what we do is we extract messenger RNA at a given time and space. That means for a specific cell at a given conditions here, and then you compare with other things here. So here a normal cell is there, a cancer cell is there. The goal is to see whether the gene expression is different in these both cases. They are cultured, 2 different cells are cultured, and then RNA is isolated, and this RNA should be converted to cDNA, the copy DNA or complementary DNA, and then the copy DNA is labeled with some kind of coloring agents such as fluorescent probes. So we can use green fluorescent for normal cells DNA and red fluorescent for cancer cell DNA, and then this DNA is hybridized to a pre-designed DNA glass slides called as DNA chips or microarray chips, and this DNA is incubated or hybridized with the microchips, and based upon the binding, the amount of binding of normal DNA or cancer DNA, the colors can be measured through laser technologies, and the data can be visualized as pixel dots of different colors, gray, yellow, red and green here. Here the green color indicate that at this spot normal cells DNA is binding more, or a specific gene is expressing more at this point here, and and here in this case cancer's related gene is expressing more. Yellow indicate that both are equally expressing, and grayish color indicate none of them are expressing or none of them are binding here. So pre-derived or pre-known information of the microchip can be used to tell the part of DNA or the part of gene which is expressing here. So using this simple image, you can tell about the progression of the disease. Basically, this image is converted to a tabular data, and this tabular data is analyzed using bioinformatic algorithms through artificial intelligence tools or data mining tools here. So functional analysis, currently rapidly growing technologies with the surgence of deep learning and machine learning technologies here. So the applications of bioinformatics